Hello students. Hello students. So today in this NEET revision series, we are going to revise a very high weighted chapter of botany, that is sexual reproduction in flowering plants. So flowering plants, they are also known as angiosperm. First, before starting with chapter sexual reproduction in flowering plants, I want to discuss a general life cycle of these flowering plants. Okay. So in life cycle of flowering plants, they are having two phases. First phase is sporophyte, second phase is gametophyte. Sporophyte is always diploid. The main plant body in case of these flowering plants or angiosperm plants, that main plant body is sporophyte. That sporophyte going to give rise to gametophyte. Again, this gametophyte going to give rise to sporophyte, that is main plant body. So sporophyte form gametophyte, gametophyte form sporophyte. This process is known as alternation of generation. So in case of flowering plants, the main plant body is sporophyte. It has something for what? Sexual reproduction. The main plant body which we see is sporophyte. This sporophyte has flower for sexual reproduction. So flower, what is flower? Flower is the modified stem. It is the sex reproduction site in case of flowering plants. Stalk of the flower is known as pedicel. Above this pedicel there is a swollen structure that is known as thalamus. To this thalamus all the floral parts, all the floral whorls, they attach. This green part individually it is known as sepal, and whorl of these sepals is known as calyx. That red color part is known as petal, and whorl of these petals is known as corolla. Then male reproductive part and female reproductive part. In case of flowering plants, it is the male reproductive part is stamen, and whorl of these stamens is known as androecium. Then female reproductive part. Female reproductive part in case of these flowering plants is carpel, and whorl of these carpel is known as gynoecium. Sporophyte. How it formed gametophyte? As the name suggests, spore. Sporophyte, first it will going to form spore. In case of flowering plants, there are two different types of spores, male spore and female spore. Male spores, they are produced by male reproductive part, that is stamen. Stamen first it will going to produce male spores, and male spores are known as microspores. Then female spores, megaspores, they are formed by female reproductive part, carpel. Sporophyte, the main plant body, it is always 2n, diploid, having two sets of chromosomes. This sporophyte, when it forms spores, these spores they are haploid. Then by which division sporophyte going to form spores? Sporophyte is 2n, whereas spores they are n. Sporophyte forms spores with the help of meiosis division. Now what is meiosis division? In meiosis division, any parental cell divides into 4 daughter cells. If parental cell having 2n number of chromosomes, then after meiosis division, daughter cell having half the number of chromosomes. So sporophyte forms spores. Then these spores further develops into gametophyte. Microspores, male spore develop into male gametophyte. Megaspores, female spores, they are developed into female gametophyte. Male gametophyte in case of these flowering plants is pollen grain, whereas female gametophyte is embryo sac. These spores are haploid. Gametophyte is also haploid. Then by which division these spores differentiated into gametophyte? By mitosis division. What is mitosis division? In case of mitosis division, any cell divides into two daughter cells, and both parental cell as well as daughter cell having same number of chromosomes in case of mitosis division. This part of the life cycle completes. Sporophyte going to form gametophyte. Sporophyte first forms spores. Male spores are microspores. Microspores are produced by male reproductive part, that is stamen. Female spores are megaspores. Megaspores, they are produced by female reproductive part, that is carpel or gynoecium. Then these spores, they are mitotically differentiated or develops into gametophyte. Male gametophyte is pollen grain and female gametophyte is embryo sac. Then second part of the life cycle. This gametophyte going to form sporophyte now. As the name suggests, gametophyte. So this gametophyte first they are going to form gamete. Male gametophyte pollen grain going to form male gamete. Female gametophyte embryo sac going to form female gamete. By which division these gametophyte which are haploid going to form these haploid gametes? Same mitosis division, as both daughter cell and parental cell having same number of chromosomes. Then what happens? Both male and female gamete they will get fused together. After fusion, zygote is formed. Zygote is diploid. Then that diploid zygote develops into the sporophyte. Zygote is 2n, sporophyte is also 2n. This zygote develops into sporophyte, which is main plant body in case of flowering plant. So this is about life cycle of flowering plant. What happens? Sporophyte going to form gametophyte going to form sporophyte. This process is known as alternation of generation. Sporophyte, it has flower for sexual reproduction. Flower has male and female reproductive parts. Male reproductive part is stamen, female part is carpel. Both these male and female parts first they are going to form spores. Male spores microspores, female spores megaspores. Then they develops into gametophyte. Gametophyte has haploid. Gametophyte is haploid. Male gametophyte is known as pollen grain and female gametophyte is known as embryo sac. Then these gametophytes further give rise to gamete. Both the gamete will get fused together. As a result, zygote is formed, and that zygote again develops into the main plant body, which we see in case of flowering plant. Okay. So first we are going to start with male reproductive part. How that male reproductive part form microspores? First, let's have a discussion about flower. Flower is the main reproductive site in case of flowering plant. It is modified stem. Okay. Top of the flower is known as pedicel. Above this pedicel, there is a swollen structure that is known as thalamus. Then green part 1 is known as sepal and whorl of these sepals is known as calyx. Then petals and whorl of these petals they are known as corolla. Then male productive part is known as stamen and this whorl is known as androecium. Then female reproductive part it is known as carpel and whorl of these carpels is known as gynoecium. So first we are going to start with male reproductive part that is stamen. Okay. So stamen basically it has two parts. This is the long thin stalk. That stalk of filament, stalk of stamen is known as filament. Above this filament there is bilobed structure, two lobes. That bilobed structure is known as anther. Thin stalk is known as filament. Above that thin stalk there is a bilobed structure that is known as anther. Now, this end of filament is known as proximal end. Proximal end of that filament is attached to what? As we discussed, above pedicel there is a swollen structure that is known as thalamus. So proximal end of this filament attached to the thalamus or that proximal end is also attached to, it can also attach to petals. If the proximal end of filament is attached to petal, then that condition is known as epipetalous condition. Now, if this end is proximal end, then opposite to proximal end, which end is present? Distal end. Distal end of this filament is attached to this terminal structure, bilobed structure that is anther. Stamen, it is the male reproductive part. It has two parts. It consists of thin filament and above that filament there is a terminal bilobed structure that is known as anther. Proximal end of the filament is attached to thalamus or that proximal end can also attach to petals. If that proximal end attached to petals, then that condition is known as epipetalous condition. Distal end attached to bilobed anther. Okay. So now what if I cut 3D section of that bilobed anther and I want to see it from the top. What is present inside that bilobed anther? So we'll discuss about that in next slide. Okay. This is filament. This end of filament is distal end. And that distal end attached to bilobed anther. Now, these are two lobes of anther. What happens? These two lobes, they are connected together with the help of tissue, that tissue is known as connective tissue. This connective tissue connects two lobes of anther. Then when we see one lobe, in case of one anther lobe, what is present? Thecas are present. One lobe has two theca. This is one theca, this is another theca. Second lobe also has two theca. This is one theca, this is another theca. Each lobe is dithecous. That's why anther is known as dithecous. In which point of view it is known as dithecous? One lobe has two theca. That's why anther is dithecous. Now, inside each theca, what is present? That yellow color one. Inside each theca, microsporangia is present. Microsporangia. Inside this microsporangia, what is going to form? Male spores are going to form inside this microsporangia, and male spores, as we already discussed, they are microspores. So where it is present? Inside each theca, there is microsporangia is present. This microsporangia is present towards the corner side of each theca. So total how many microsporangia are present in one anther? One anther is having two lobes. Inside each lobe there is two microsporangia. So two lobes having four microsporangia. Two lobes, they are connected together with the help of connective tissue. Now, these microsporangia, inside these microsporangia, microspores are formed. Then microspore, they develops into male gametophyte, that is pollen grain. So, these microsporangia, they further develops into pollen sac, and pollen sac contains lots of pollen grains, that is male gametophyte in case of flowering plants. Inside this microsporangia, we are going to form microspores. There is a longitudinal groove runs along the lengthwise of these theca, and that longitudinal groove separates these two theca. This longitudinal groove going to separate two thecas, and these are line of dehiscence from where anther dehisces to release the male gametophyte, that is pollen grain. Now transverse section of anther. Anther is having two lobes. These two lobes, they are connected together with the help of a tissue that is known as connective tissue. Each lobe has two theca, one theca, second theca. Each lobe has two theca, one second. Both each lobe has two theca, and inside each theca, towards the corner side, what is present? Microsporangia is present. Now, this microsporangia is surrounded by how many wall layers? 1, 2, 3, 4. This microsporangia is surrounded by 4 wall layers. What are these wall layers which surround that microsporangia? Let's start from outer side. Outermost layer is known as epidermis. Then second layer is known as endothecium. Then third layer is middle layer. And fourth one layer is known as tapetum. There are four more layers which surround the microsporangia. That sequence of these four layers from outer side to inside is epidermis, endothecium, middle layer, and last one is tapetum. What about their sequence from inside to outer side? Tapetum, middle layer, endothecium, epidermis. This is about transfer section of anther. Now let's discuss something about features of these four wall layers which surrounds the microsporangium. First layer is known as epidermis, then endothecium, then middle layer, then last one is tapetum. Epidermis, it is a single layer. Endothecium is also single layer. Last one, tapetum, is also single layer. Then what about middle layer? Middle layer is multi-layered. Out of four wall layers of microsporangia, which layer is multi-layer? Middle layer is multi-layered. The outer three wall layers, they helps in protection of what? As they surround microsporangia, so they helps in protection of microsporangia. Second one, they helps in dehiscence of anther to release what? Pollen grain, and pollen grain is the male gametophyte here. What about last layer, that is tapetum? Tapetum is single layered. This tapetum has dense cytoplasm. This layer is nutritive layer. It contains lots of nutrition. This layer, tapetum layer, going to provide nutrition to the developing pollen grains which develops inside this microsporangia. Outer three layers, they help in protection and dehiscence of anther to release pollen grains. Inner one helps in providing nutrition to the developing pollen grains. This layer also secretes an enzyme, and that enzyme is callase. What is the role of this callase? We'll see in upcoming slides. One more characteristic feature of this layer is it is multinucleated. Why this tapetum layer is multinucleated? See, when this tapetum layer undergoes division, cells of tapetum layer undergoes division, then what happens? As we know, when cell undergoes division, first step is karyokinesis. And what is karyokinesis? Karyokinesis is the division of nucleus. Second step is cytokinesis. In case of these tapetum cells, what happens when cell undergoes division? Yes, karyokinesis occurs, but there is no cytokinesis. That's why this tapetum layer is multinucleated, because during its division only karyokinesis takes place. This is about four wall layers which surround the microsporangia. This is connective tissue which connects two lobes of anther. Then inside each theca, what is present? That is microsporangia. Microsporangia is further surrounded by four wall layers. Outer one is known as epidermis, then endothecium, third one is middle layer, and the layer which surrounds this microsporangia is tapetum. Outer three wall layers, they help in protection of this microsporangia as well as dehiscence of pollen grain. Now, Which layer provides nutrition to the developing pollen grains? That is tapetum. When anther is in young condition, then what happens? There are some cells which occupies center of what? Microsporangia. In which condition? When anther is in young condition. When anther is young, then cells of some tissue occupies center of this microsporangia, and these cells are known as sporogenous tissue cells. These sporogenous tissue cells, they occupy center of the microsporangia. These cells, they are compactly arranged cells. They are same kind of cells. That's why they are known as homogeneous cells. Eh, What about the ploidy of these sporogenous tissue cells? As they are part of male reproductive part stamen, and stamen is the part of flower, flower is the part of sporophyte, and sporophyte, we know that sporophyte is 2n. So these cells, they are also 2n diploid. When anther is in young condition, then these cells, they occupy center of the microsporangia, and these cells are known as sporogenous tissue cells. They are compactly arranged cells. They are same kind of cells. That's why they are homogeneous cells, and their ploidy is 2n. Now, what happens when anther starts to mature or develop? See, this is the condition when anther is young. Sporogenous tissue cells, they occupy center of the microsporangia. Now, what happens when anther starts to mature? When anther starts to mature, then these same cells, sporogenous tissue cells, which are diploid cells, these cells undergo which division? Sporophyte going to form spores by which division? Meiosis division. So these sporogenous tissue cells, they undergoes meiosis division. As a result, what is going to form? Four microspores are formed by these sporogenous tissue cells. Initially, what happens? These four microspores, they are arranged together. They are arranged in cluster of four, and these four microspores, they are held together with the help of some wall. That wall is known as callose wall. And as we discussed, the last layer, tapetum, innermost layer, secretes some enzyme, which is that enzyme callase. That callase going to break that wall, callose wall. Sporogenous tissue cell, when anther mature, it undergoes meiosis division. As a result, microspore tetrad is formed. These microspores, they are arranged in clusters, and they are held together with the help of the wall. That wall is known as callose wall. Now, as each and every cell of this sporogenous tissue has the capability to give rise to microspores. That's why this cell is also known as potential pollen. This sporogenous tissue cells is also known as microspore mother cell. What are the other names of that sporogenous tissue cell? They are also known as microspore mother cell, as they are each cell of sporogenous tissue has the capability to give rise to microspores. That's why it is also known as potential pollen. Now, what is microsporogenesis? Sporogenous tissue cell undergoes meiosis division. As a result, microspores are formed. That process of formation of microspores is known as what? Microsporogenesis. So, can you please tell me, if sporogenous tissue cell is diploid 2n, then What is the ploidy of these microspores which are formed after meiosis division? They are haploid. Now, when further maturation occurs, then what happens? That callose wall is break down by the enzyme callase, and this enzyme callase is secreted by tapetum. So now this concept is clear that what is the role of callase. When anther further mature, one more thing also takes place, that is dehydration. On further maturation, dehydration takes place also, that microspore tetrad is dissociated, callase enzyme dissolves that callose wall. As a result, what happens? Each microspore is formed, and that microspore further differentiated to form pollen grain, that is male gametophyte. Now, we just completed with one portion of our life cycle of angiosperm plant. Sporophyte, it has male part is stamen, the thin stalk is known as filament. Above this there is bilobe structure that is known as anther. When we cut the transverse section of that anther, then what is present? These two lobes, they are connected together with the help of some tissue that is known as connective tissue. Inside each lobe there are two thecas. These two thecas, they are going to, inside these two theca, what is present? Microsporangia occupies the towards the corner side of these thecas. Then what happens? Microsporangia is surrounded by four wall layers: epidermis, endothecium, middle layer, last one is tapetum. Then microsporangia, when anther is in young condition, then what happens? When anther is in young condition, cells of sporogenous tissue, compactly arranged cells, they occupies the center of microsporangia. When anther start to develop, then what happens? These sporogenous tissue cells, they undergoes meiosis division, and as a result of it, what is formed? Microspore tetrad is formed. That microspore tetrad, on further maturation of anther, dehydration takes place and callose wall dissolved by the callase enzyme. Then what happens? Each microspore develops into the pollen grain, that is male gametophyte. How that microspore develop into pollen grain? By mitosis division. Now we will see how that microspore develops into pollen grain. Okay, so before starting with that, let's draw a flowchart of the event. Okay, initially what is there? Sporophyte, which is 2n. That sporophyte has male reproductive part, and that male reproductive part is known as stamen. That stamen has two structures. What are these? Stalk is known as filament. Above this filament, there is a terminal bilobed structure that is known as anther. Now, inside anther, anther is bilobed. Each lobe has two thecas, and inside each theca towards corner side, what is present? Microsporangia. Then what happens when anther is in young condition? In young stage, what happens? Sporogenous tissue cells, they occupy center of the microsporangia. What about ploidy of these cells? They are also 2n. Then what happens when anther starts to mature? Then each cell of this sporogenous tissue undergoes meiosis division, and as a result of it, what is formed? Microspore tetrad is formed. Now, can you please tell me some another name of these sporogenous tissue cells? They are also known as microspore mother cell as well as potential pollen. Then what happens? On further maturation and dehydration, what happens? Microspore tetrad dissociates and each microspore develops into pollen grain, which is the male gametophyte. Till now we discuss about this thing. So as meiosis division takes place, then what happens? Ploidy level changes from 2n to n. Now we are going to start with what is microspore structure and how that microspore develops into pollen grain. How that microspore matures to become pollen grain. How mitosis division takes place inside these microspores. Initially, what is present? Microspore after meiosis division. Okay, microspore, what is the polarity of that microspore? It is haploid as it is a result of meiosis division. Now, that microspore has two wall layers. Microspore is surrounded by two wall layers. Out of these two wall layers, outer layer is known as exine, inner layer is known as intine. Outer layer is made up of what? That outer layer is made up of sporopollenin. Then what about inner wall layer? Inner wall layer is made up of cellulose and pectin. Outer wall layer is thick. Inner wall layer intine is thin. Now there are some properties of this sporopollenin. Sporopollenin is one of the most resistant organic material. One of the most resistant organic material. This sporopollenin can withstand at high temperature, alkaline, acidic, any conditions. Even no enzyme can degrade this sporopollenin. That's why the layer is thick. Now, how can you please tell me by saying this diagram, out of these two layers, which layer is continuous and which layer is non-continuous or discontinuous? As we see, the inner layer is continuous layer. Outer layer that is made up of sporopollenin, that layer is discontinuous layer. Why this layer is discontinuous? Because where in these areas where this outer layer is absent, that aperture is known as germ pore. What is the role of that germ pore? When this microspore matures into pollen grain, then after landing on stigma, this pollen grain needs to form pollen tube. So this germ pore is that aperture where sporopollenin is absent, and this germ pore helps in guiding the formation of pollen tube. Pollen tube going to form from one of the germ pore. Because of this sporopollenin, pollen they can also preserved as fossils. Out of these two layers, which layer exhibit fascinating pattern and design? Same exine layer. It exhibits fascinating pattern and design. Because of this sporopollenin absent in some areas, and these apertures are known as germ pore. Now, what is the diameter of that pollen grain? It is about 25 to 50 micrometers. As we discussed, microspore has two wall layers, intine, exine. Now next step is how that microspore matures to become pollen grain. Sporophyte already forms spores, male spores are microspores. Now how these microspores develop into the male gametophyte pollen grain? That by which division? That is occur by mitosis division. So we will going to see how that microspore matures into pollen grain by mitosis division. See, initially what happens? Microspore, then vacuole, they occupies most of the part of this cell. As a result, what happens? The nucleus it shifts towards the periphery. Then what happens? The asymmetric spindle formed. Due to formation of these asymmetric spindle, that cell divides into two unequal cells. What happens? Initially microspore is there. Vacuoles they occupy most of the part of this microspore. As a result, what happens? The nucleus is shift towards the periphery. Then due to formation of asymmetric spindle, this leads to unequal division of that microspore, and due to that unequal division, one cell is large cell, another cell is smaller one. Large cell is known as vegetative cell, and smaller cell is known as generative cell. Now we are going to start with some characteristic features of these two cells. Smaller cell is known as generative, larger one is known as vegetative. How these cells are formed? Due to unequal division of microspore. How that unequal division occur? Because of asymmetric spindles. Why these asymmetric spindle form? Because of vacuoles, they occupy center or most of the part of the microspore. As a result, nucleus shift towards the periphery. Vegetative cell. This cell is large, generative cell is small. Okay, one difference clear. Second thing, generative cell, as the name suggests, generate. These generative cells, they are going to generate something. What is that something? These generative cell going to generate male gametes. We'll see how that takes place. Generative cell, what is the shape of that generative cell? These cells, they are spindle in shape. These vegetative cells, they contains reserve food material. They provide nutrition to this generative cell. This vegetative cell has irregular shaped nucleus. Now, out of these two cells, which cell float in the cytoplasm of which cell? This smaller cell, generative cell, it appears that it floats in the cytoplasm of large cell, that is vegetative or reserve. It contains reserve food material. What happens in angiosperms? Out of 100%, almost in 60% of angiosperms, that mature pollen grain which is having in two cell stage, vegetative cell and generative cell, that pollen grain shed at this two cell stage. Then what about remaining 40%? In remaining 40%, what happens? That generative cell undergoes mitosis division and it forms two gametes. Then total how many cells are there? Two gametes plus this vegetative cell. Is this clear? Okay. Now, pollen as nutritional source, as well as it creates some problems like it causes allergy. Pollen grains, they are rich in nutrients. Tablets of pollens, they are used as food supplement. Pollen products in the form of tablets and syrup, they are available in the western countries market. Pollen consumption increase or enhance the performance of athletes as well as race horses. Now, what about the demerits? Pollen causes some allergy and bronchial afflictions like asthma and bronchitis. One famous example of carrot grass. Scientific name of that carrot grass is Parthenium. This Parthenium is not native of India. This Parthenium came into India as a contaminant with imported wheat, and this Parthenium caused pollen allergy. These are about nutritional and demerits of this pollen grain. Last topic in case of male reproductive part is pollen grain viability. What is pollen grain viability? The period for which pollen grains retain its ability after shedding and transfer on the stigma to form the pollen tube. It is the period for which pollen grain retain the ability to germinate on landing at the stigma. This pollen viability is further depends on some factors. These factors are temperature and humidity. In case of some cereals like wheat, rice, pollen grain lose its viability time period within the 30 minutes or half an hour. In some families like Rosaceae, Leguminosae and Solanaceae, in these families what happens? pollen remain viable for few months. Now, are these pollen grains can be stored? Yes, pollen grains they are stored in the liquid nitrogen, and temperature of liquid nitrogen is minus 196 degrees Celsius. This is about the male reproductive part. Now we are going to start with female reproductive part. Before starting with that, let's try to solve some questions. Okay, these are NCRT lines. Let's recall these lines. First, site of sexual reproduction is site of sexual reproduction is flower, which changes leads to differentiation and development of flower primordium. Some structural and hormonal changes takes place which leads to formation of floral primordium. Third, proximal end of filament is attached to proximal end, it can attach to thalamus or it can also attach to petal. Next, typical angiosperm anther is it is bilobed. Microsporangia develop further to become what? They develop further to become pollen sac, which contains lots of pollen grains. Next question. Sequence of wall layers surrounding microsporangia from inside to outside. Okay. Inner one is tapetum, then middle layer, then endothecium, and outer one is epidermis. Next, cells of sporogenous tissue is homogeneous or heterogeneous? Are they made up of same kind of cell or different kind of cells? They are homogeneous. These can come as out of these statements. Which one is correct, incorrect, statement 2, statement 1 based question. Now, ploidy of. We need to find out ploidy level. Sporogenous tissue. It is the part of sporophyte and we know sporophyte is diploid. Microspore tetrad. We know microspore tetrad is formed by meiosis division. So this is haploid. Sporophyte, it is the main plant body that is diploid. Potential pollen is also known as sporogenous tissue cell or microspore mother cell. It is also 2N. Endothecium is the wall layer which surrounds microsporangia. So it is also part of sporophyte. Male gametophyte is haploid. Generative cell and vegetative cell. These are part of pollen grain. So these are also haploid. Microsporangium, that is part of sporophyte. It is 2N. Pollen grain is the male gametophyte. It is N. Outermost layer exine. Exine is the part of microsporangia. So it is Exine is the part of microspore, so it is N. Next question. This continuous layer of pollen grain is made up of, that means inner layer or outer layer. This continuous layer is exine, and exine is made up of sporopollenin. Now, what is germ pore? Germ pore is the aperture where sporopollenin absent, and that germ pore helps in guiding the formation of pollen tube. Next. I'm not going to fill these, you fill them yourself. The two anther lobes, they are attached to each other by two anther lobes. They are attached with the help of some tissue that is known as connective tissue. Sporogenous tissue undergoes meiosis division to form what? Microspores. As the anther mature and rehydrate, the microspore dissociate from each other and develops into pollen grain. Is this statement true or false? Instead of rehydrate, dehydration is there. Pollen grain wall is single layer. Is this statement true or false? That is double layer. Hard outer layer is exine, and that exine is made up of sporopollenin. Part of exine where sporopollenin is absent, that is germ pore. Mature pollen contains two cells. These cells are vegetative cells and generative cells. Dash floats in the cytoplasm of dash. Vegetative cell floats or generative cell floats? Smaller one is generative cell. Generative cell floats in the cytoplasm of larger one cell, that is vegetative cell. Now, which statement is incorrect? We need to find out incorrect statement. Intine is inner wall of pollen grain and exhibit fascinating array of pattern and design. Yes, intine is the inner wall layer, but exine exhibits fascinating array of pattern and design. So this statement is incorrect. The mature pollen grain has two cells. Bigger is vegetative, smaller one is generative, which floats in cytoplasm. This statement is true. Carrot grass, it causes pollen allergy. Scientific name is Parthenium. Pollen grain of pea and rose, they maintain their viability for months. Yes. Next question. Pollen grain. Pollen grain, it represents the female gametophytic phase of plant. It is the male gametophyte. Wrong. Pollen grain can cause several allergies like asthma and bronchitis. D is correct. Pollen grains, they are rich in nutrient and are used as food supplements. Yes. They can be stored in liquid nitrogen, and we know the temperature of liquid nitrogen is 196 degrees Celsius. Out of these B, C, D, only B, C, D are correct. So correct option is D. In some members of which of the following pair of families pollen grain retain the viability for months after release? We know three families are there: Rosaceae, Leguminaceae, and Solanaceae. So which option is correct? Rosaceae and Leguminaceae. Next question. An organic substance which can withstand environmental extreme and cannot be degraded by any enzyme, that substance is sporopollenin. Outer layer is made up of this sporopollenin only. Next question. Male gametophytes in angiosperm possess. Male gametophyte. We know male gametophyte is pollen grain. And pollen grain has what? Vegetative cell plus two male gametes. So out of these, which one is correct? Two gametes and vegetative cell. Option C. Which one of the following statement is correct? Sporogenous tissue is haploid. No, sporogenous tissue is 2N, diploid. Endothecium produce the microspore. Sporogenous tissue cell undergoes meiosis division. It will produce microspore. Tapetum nourishes the developing pollen grain. Yes, tapetum is the nutritive layer. Hard outer layer is made up of cellulose and pectin. No, that hard outer layer is made up of sporopollenin. Next. Which one of the following statement is incorrect? Stored pollen in liquid nitrogen, they can be used in crop breeding programs. Yes, this statement is true. Pollen grains of many species, they can cause several allergies too. Tapetum helps in dehiscence. No, outer three wall layer helps. Hard outer layer is sporopollen. So C is incorrect. Next, size of pollen grain. Size of pollen grain is 25 to 50 micrometer. Proximal end of filament of stamen is attached to. Proximal end, it can attach to thalamus or it can also attach to petal. What about distal end? Distal end attached to bilobe anther. Next question. Pollen tablets, they are available in market for ex situ conservation? No. In vitro fertilization? No. Breeding programs? No. Supplementing food? Yes, this statement is. How many meiotic and mitotic divisions are required for the development of 16 male gametes from microspore mother cell in flowering plant? So you have 2 minutes to solve this question. Okay, we'll discuss. What is given? How many meiotic and mitotic divisions they are required for the development of 16 male gametes? Okay, let us suppose one microspore mother cell is there. One microspore mother cell, it undergoes one meiosis division, then it forms four microspores, and four microspores, they are mitotically. Four microspores undergoes, if we consider one microspore, then one microspore undergo how many mitotic division to form pollen grain? Tell me. One microspore undergoes one mitotic division to form pollen grain, and again that pollen grain undergoes one more mitotic division to form how many male gametes? Two male gametes. And in question, we are supposed to find out for 16 male gametes. When two male gametes are formed, for the formation of two male gametes, what is required? How many mitosis and meiosis? For two male gametes, one meiosis and two mitosis. Then we need to find out for 16 male gametes. First, find out for one male gamete, then 16 male gamete. Next question is, there are 10 flowers in one individual pea plant. In each microsporangium of every stamen of all the flowers, there are 30 microspore mother cells. We need to find out how many pollen grains are formed from that plant. Okay. Again, you have 2 minutes to solve. Okay, let's discuss. There are 10 flowers in one individual pea plant. What is given? 10 flowers. In each microsporangium of every stamen of all the flowers, there are 30 microspore mother cells. And we are supposed to find out number of pollen grains formed from that plant. Okay. See, we know in case of pea plant. Let's recall morphology in flowering plants chapter. Pea plant has how many stamens? 10 stamens. Diadelphous condition we already discussed. Pea plant has 10 stamens. So we know one flower has 10 stamens. And what is given? 10 flowers. 10 flowers has 100 stamens. Okay, it's clear. Then what is given? In each microsporangium of every stamen, there are 30 microspore mother cells. We know in one stamen there are 4 microsporangia. Then what about these 100 stamens? 100 stamens has 400 microsporangia. As we know, one microsporangia has how many microspore mother cell given? In each microsporangium of every stamen, there are 30 microspore mother cells. One microsporangia, how many mother cells are given? 30. Then what about these 400 microsporangia? 400 into 30. That is 12,000 microspore mother cell. And we already know one microspore mother cell forms how many pollen grains? It undergoes one microspore mother cell undergoes one meiosis division and forms four microspore tetrad. They develops into four pollen grains. Then what about this number of microspore mother cells? 12,000 microspore mother cells, they will going to form how many pollen grains? 48,000 pollen grains. So out of these four options, which option is correct? Option number 1. One thing we need to remember is in case of pea plant, they are having 10 stamens. So 48,000 is the correct answer. Now, next question. Assertion reason based. Cells of the tapetum, they possess dense cytoplasm and generally they have more than one nucleus. Yes, they have one more than one nucleus and they are having dense cytoplasm. Assertion is true. Reason. Presence of more than one nucleus in the tapetum increases the efficiency of nourishing the developing microspore mother cell. Yes, tapetum, they are going to provide nutrition to the developing pollen grains. So presence of more than one nucleus, they helps or enhance that thing. So reason is the correct explanation of assertion here. Correct option is both A and R are true and R is correct explanation of A. Now can we switch to our next topic, that is female reproductive part. Yes. So our next topic is female reproductive part, that is pistil. What is gynoecium? Gynoecium, it is the whorl of the female reproductive part, that is carpel. Individual unit of that gynoecium is known as carpel. Okay. This gynoecium, it can be monocarpellary and multicarpellary. Single carpel can be present and more than one carpel can be present. If multicarpellary situation is there, more than one carpels are present, then what happens? They can be fused together or they can be free. So If more than one carpel is present and these carpels they are fused together, then that condition is known as syncarpus. Example of syncarpus condition is papaver. Now, can you please tell me two more examples of syncarpus condition from the chapter Morphology of Flowering Plant? Mustard and tomato. They are also example of syncarpus in which pistils are fused together. Next is apocarpus. What is apocarpus? When these pistils they remains free, then that is known as apocarpus condition. Example is Michelia. Again, two more examples are given in morphology of flowering plants. These are rose, lotus. Mark these as very important. Next, parts of pistil of female reproductive part. It has three parts: stigma, style, ovary. Three parts: stigma, style, ovary. What is stigma? Stigma it acts as the landing platform for male gametophyte, that is pollen grain. Stigma it acts as the landing platform for the pollen grain. Style, beneath this stigma, this stalk is present, that is style. Third part is ovary. Ovary is the basal bulge part. The cavity in space, the space which is present inside this ovary, that space is known as ovarian cavity, and another name of ovarian cavity is locule. Inside this locule there is some structure is present, that is known as placenta. And what is placentation? Arrangement of placenta inside the ovary is known as placentation. There are five different kinds of placentation given in morphology of flowering plant. Placenta is present inside the ovarian cavity or locule. This placenta helps in formation of megasporangia. In case of male part, that is microsporangia. Here it is megasporangia, ovule or megasporangia. What is placenta? Megasporangia or ovule arise from the placenta. It is present inside the ovarian cavity or locule. This figure shows papaver in which syncarpus condition is there, and this is about Michelia where apocarpus condition is seen. Now, megasporangium and ovule. In case of angiosperm plants, anatropous ovule is present. What is anatropous ovule? It is inverted ovule. What happens in case of inverted ovule? Micropyle and funicle, they are close, they lie close to each other. Okay. As we discussed, inside ovarian cavity, what is present? Placenta, and this placenta helps in formation of megasporangia and ovule. The stalk with the help of this ovule body attaches to placenta. That stalk is known as funicle, and this ovule body fuses with this funicle in a special region that is known as hilum. So hilum is the junction between ovule body and funicle. Placenta, there is a stalk. With the help of this stalk, this ovule body attaches to placenta. Then this ovule body fuses with that funicle inner region. That region is known as hilum. Now, there are two protective coverings. These are known as outer integument and inner integument. These integument layer, they are absent in some specific region. That region where there is no integument, that is known as micropyle end. Opposite to this micropyle end is chalazal end, and chalazal end is known as base basal part of ovule. Placenta, funicle, hilum, outer integument, inner integument, micropyle, and where integuments are absent. Opposite to it is chalazal end. Now, these integuments encircle inside some tissue. That tissue is known as nucellus. Nucellus is the nutritive tissue. One cell of that nucellus. Can you please tell me the ploidy level of nucellus, as it is part of sporophyte? Now here we are discussing about female part. So nucellus is 2n. One cell of that nucellus which is present towards the micropyle end. That one cell acts as microspore mother cell. In case of male reproductive part, we observed that each and every cell of sporogenous tissue has the capability to give rise to microspores. But here what happens? Only one cell of this nucellus tissue. Which cell of that nucellus tissue? That is present towards the micropyle end. That cell grows in its size and that cell acts as megaspore mother cell. That means that cell going to undergo meiosis division to form female spores, that are megaspores. So this is about ovule, and basal part is which is present opposite to micropyle. Now, ovule arise from the placenta. What is funicle? Funicle is the stalk with the help of ovule body attaches to the placenta. Hilum, ovule body fuses with funicle in the particular region that is known as hilum. What is the junction between ovule and funicle? Funicle and ovule, that is hilum. Nucellus, enclosed inside the integument. This is a reserve contain. This nucellus contains reserve food material, nutritive tissue, and ploidy of that is 2n. Integuments. These are protective covering. There are two integuments, outer and inner. Next, micropyle. Micropyle is that end where these integument layer are absent. Next is chalaza. Opposite to the micropyle, chalaza is present, and this chalaza is known as basal part of ovule. Next, basal part of ovule is chalazal. What is embryo sac? Embryo sac is the female gametophyte which is going to form inside the nucellus. One ovule, okay. Ovule can be present one in an ovary, or it many ovules are also present in an ovary. Example of one ovule in an ovary is wheat, paddy, mango. What about many ovules in ovary? Examples are papaya, watermelon, and orchid. Next, this is ovule, funicle, hilum, integuments, micropyle, chalazal, which is a base. nucleus is 2n. I repeated the same statement that nucleus tissue, one cell of that nucleus, which cell that is present toward micropyle end. That end is micropyle end. So one cell which is present towards micropyle end, that cell acts as megaspore mother cell, MMC. What about ploidy of this cell? This cell is 2n, single cell of nucleus which is present towards micropyle and it acts as MMC, megaspore mother cell. What are the properties of that single cell which acts as megaspore mother cell? That cell is large cell, it has dense cytoplasm, it has prominent nucleus. This megaspore mother cell undergoes meiosis division, and we already know when this megaspore mother cell undergoes meiosis division, as a result of it, what are formed? Microspores are formed. How many microspores are formed? Four microspores are formed. These, sorry, four megaspores are formed. Megaspore mother cell undergoes meiosis division. As a result of it, megaspores are formed. How many megaspores are formed? Four megaspores are formed. And what about the ploidy of these megaspores? These are haploid. So we see in case of male reproductive part, when four microspores are formed, these all the microspores that differentiated into male gametophyte, that is pollen grain. But here what happens? Out of the four megaspores, three megaspores degenerate. Which three megaspores degenerate? Those megaspores which are present towards the micropyle end, they degenerate. Only one megaspore remains functional. That means this functional megaspore going to form female gametophyte, that is embryo sac. And from this megaspore mother cell, formation of megaspores is known as megasporogenesis. Megasporogenesis, out of four megaspores, three megaspore degenerates which are present towards the micropyle, and only one megaspore is functional, that is present towards the chalazal, and that one functional megaspore now develops into female gametophyte, that is embryo sac. This is about anatropous. This diagram is about anatropous ovule. Then micropyle and nucleus tissue. One cell of that nucleus acts as megaspore mother cell, which is having large cell, dense cytoplasm, prominent nucleus. Then what happens? This megaspore mother cell undergoes meiosis division. After meiosis, what happens? During meiosis, after meiosis 1, two cells are formed. Then tetrad is formed after meiosis 2. This is about megasporogenesis. Next. Now, what is monosporic development? Functional megaspore. Which is that functional megaspore? Is that present towards chalazal or micropyle? That megaspore is present towards the chalazal. Functional megaspore present towards chalazal. This functional megaspore develops into embryo sac, that is female gametophyte. This complete process of formation of female gametophyte from one functional megaspore, that is known as monosporic development. Embryo sac formation from the single megaspore. Now we will discuss how that One functional megaspore form embryo sac. By which division? Mitosis division. See, functional megaspore, micropyle, chalazal. Okay, this functional megaspore develops into female gametophyte embryo sac. By which divisions? Mitotic division. This functional megaspore it undergoes this functional megaspore undergoes first mitotic division, first mitosis, and that mitosis is free nuclear division. That means karyokinesis is not followed by cytokinesis immediately. That functional megaspore undergoes mitosis division. As a result of it, how many nucleus are formed after karyokinesis? Two nucleus cells. Two nucleus, they move towards the opposite poles, chalazal and micropyle. Then what happens? Second mitotic division occurs. That second mitotic division also free nuclear. That means only karyokinesis takes place. Cytokinesis not followed that immediately. During second mitotic division, from these two, how many nucleus are formed? Four. That stage is known as four nucleate stage. Then third mitotic division occurs. After third mitotic division, how many nucleus are formed? Eight nucleus are formed. Now what happens? Cell wall formation takes place after this. And that cell wall formation takes place around only 6 of the 8 nucleus. These are 8 nucleus. Only out of these 6 nucleus they are surrounded by the cell wall. We will see in next slide what is the complete mature embryo sac. Yes. Distribution of cells inside the embryo sac. 8 nucleus were there. Out of these 8, only 6 nucleus are surrounded by the cell wall. Three nucleus or three cells which are present towards the chalazal end. These cells which are present towards chalazal end, they are known as antipodal. Antipodal cells, they are present towards the chalazal end. Then what about micropyle end? Towards micropyle end, two cells are known as synergid. Third one is known as egg, and synergid plus egg, they are formed egg apparatus. Egg apparatus. What is egg apparatus? Egg cell plus synergid. These are present towards the micropyle and they form egg apparatus. Now, what about those nucleus? These two nuclei, they are known as polar nuclei. Polar nuclei, they are present inside this large cell and that large cell is known as central cell. So can you please tell me out of chalaza and micropyle which is the basal part? Chalaza is the basal part. So can I say that polar nuclei, they are present below this egg apparatus. If chalaza is the base, Then these polar nuclei, they are present below the egg apparatus. Mark this line as very important. They can give above here. So that above one is incorrect, below is correct. Now this synergid has some finger-like extensions. These are known as finger-like extensions. These are known as filiform apparatus. What is the role of filiform apparatus? These filiform apparatus guide the entry of pollen tube inside the synergid. This is about mature embryo sac. So can you please tell me that mature embryo sac having how many cell and how many nucleus? 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleus are there. What about cell? 1, 2, 3, 4, 5, 6 and large central cell. So mature embryo sac is 7 celled, 8 nucleated structure. This is about female reproductive part. We Now we are going to start with question part of this. Yes. Ovarian cavity is also known as ovarian cavity is locule. Recall, placenta is located outside the ovarian cavity. No, this statement is incorrect. Placenta is located inside the ovarian cavity. Megasporangia, they are commonly known as ovule. Megasporangia, they arise from placenta. Yes, placenta helps in formation of megasporangia. That statement is true. Example of syncarpous ovary is papaver, mustard, tomato. Single ovule present in wheat, paddy, mango. What about multiple ovules? Papaya, watermelon. Then last, ovule is attached to placenta with the help of stalk. That stalk is known as funicle. The body of ovule fuses with funicle in a region that is known as hilum. Integument, they encircle the nucleus but except at a region that is known as micropyle. Next, opposite to the micropyle, what is present? Which end is there? Chalazal end. So micropyle represent the basal part of ovule. Is this statement true or false? False. Chalazal is the basal part. Dash is enclosed within integument. Within integument, a nutritive tissue is present that is known as nucleus. Female gametophyte is also known as embryo sac. Dash have abundant reserve food material. Nucleus tissue has abundant reserve food material. And ovule generally has single embryo sac. Yes, an ovule generally has single embryo sac. This statement is true. Ovules, they generally differentiate a single megaspore mother cell from which region of the nucleus? Is it present towards chalaza or micropyle? That is present towards the micropyle. Megaspore mother cell have thin or dense? Which cytoplasm and inconspicuous or prominent nucleus? They are having dense cytoplasm and prominent nucleus. What is monosporic development? One functional megaspore going to form the female gametophyte. That is known as monosporic development. Now, ploidy of female gametophyte. Female gametophyte is N. Nucleus. Nucleus is 2N. Megaspore mother cell 2N. Functional megaspore. N. Embryo sac, it is the female gametophyte. N. Potential pollen, it is the sporogenous tissue cell. 2. N. Next. Next. How many mitotic divisions required to form female gametophyte from the functional megaspore? How many mitotic divisions? 3 mitotic divisions. And how many cell and nucleus are there? 7 cells, 8 nucleus. Are these divisions pre-nuclear? Yes, these divisions are pre-nuclear. Cytokinesis not immediately followed karyokinesis. Out of 8, how many nucleus are surrounded by cell walls? Only 6. Polar nuclei, they are situated above the egg apparatus in the large central. Is this statement true or false? Polar nuclei, they are present above or below. If chalazal is the base, that means below the egg apparatus. Cells present at micropyle end, they form egg apparatus. And egg apparatus has two parts, egg cell and synergid, two synergids. Cells which are present at chalazal end, these are known as antipodal. Next, filiform apparatus is the feature of which cell? Synergid cell has filiform apparatus which guides the entry of pollen tube. Role of filiform apparatus, it guides the entry of pollen tube. Typical angiosperm embryo sac, on its maturity, it contains 7 cells, 8 nucleated structure. This is about female reproductive part. Next, egg apparatus, it consists of synergids and egg cell. Synergids, they are having special thickening at micropyle tip. These are known as filiform apparatus. Their function is they guide the entry of pollen tube. At typical angiosperm embryo sac, at its maturity, it shows 7 nucleus, 8 cell structure. This is incorrect. 7 cell, 8 nucleus is correct. 3 cells which group together at micropyle and they are known as antipodals. No, antipodals are present at chalazal end. This is incorrect. Polar nuclei, they are situated above the egg apparatus. Again, wrong statement, they are situated below the egg apparatus. Now, ploidy. Megasporangium is ovule, it is 2N. Megaspores, they are haploid. MMC, diploid, female gametophyte, and egg, synergid, antipodals, they are part of female gametophyte only. That's why they are N. Polar nuclei and central cell, they are also N. An ovule has a single embryo sac formed from megaspore through which division? Reduction division? No. Mitotic divisions, yes. How many mitotic divisions? Three mitotic divisions. Next, embryo sac is monosporic when it develops from one of the four megaspores of megaspore mother cell, three megaspores of megaspore tetra, two functional megaspores, or the megaspore mother cell where meiosis has occurred but no cytokinesis. Only one of the four megaspores of megaspore mother cell, that is functional megaspore. Starting from the innermost part, the correct sequence of parts in an ovule are innermost. Okay. Egg, nucellus, embryo sac, integument, no. Then egg, embryo sac, nucellus, integument. Integument, they encircle nucellus. Inside nucleus, what is present? Female gametophyte. That female gametophyte has egg cell. This is correct. Next, from the statements given below, choose the option that are true for a typical flowering plant. Typical female gametophyte. It is 7 cell 8 nucleate stage. Yes, this statement is true. It is free nuclear during the development. Yes, free nuclear divisions are take place. It is situated inside the integument but outside the nucellus. No, it is present inside the nucellus only. It has an egg apparatus situated at chalazal end. Chalazal end has antipodals. So, statement 1 and statement 2 are correct, true. Functional megaspore in an angiosperm develops into female gametophyte, and that female gametophyte is embryo sac. Megasporangium is equivalent to ovule. Next, function of filiform apparatus. Filiform apparatus, they guide the entry of pollen tube, and these are the feature of which cells? Synergid cells. Filiform apparatus is the characteristic feature of which cell? Synergids. And these are present towards the micropyle end. In angiosperms, microsporogenesis and megasporogenesis involves what? Sporophyte form spores. Spores. Again the same life cycle. 2n n. Male spore formation is microsporogenesis. Female spore formation is megasporogenesis. So by which division they are going to form? Meiosis division. So which is correct option here? It involves meiosis. Next, in majority of angiosperms, reduction division occur in megaspore mother cell. A small central cell is present in the embryo sac. No, that cell is large. Egg has filiform apparatus. No, synergid has filiform apparatus. There are numerous antipodal cells. No, there are only three antipodal cells. So reduction division occurs in megaspore mother cell. An ovule of angiosperms is technically equivalent to, ovule is also known as megasporangium. So correct is C. Option C is correct. That's all. Now next topic is pollination. We'll start with that topic within 2 minutes. Okay. So next topic is pollination. What is pollination? Transfer of pollen grain from anther to stigma of pistil. But why that pollination is required? Why there is requirement of transfer of this male part to the female reproductive part? Because in case of flowering plant, male part as well as male and female reproductive part gametes, they are non-motile. That's why there is requirement of transfer of these pollen grains from anther to stigma. So that transfer takes place with the help of some pollinating agents which are going to do that pollination. So depending on the source of pollen, that pollination can be divided into three types. What are these three types? Autogamy, zytnogamy, xenogamy. What is autogamy? Transfer of pollen grains from anther to stigma of same flower. transfer of pollen grains from anther to stigma of same flower. That is known as autogamy. What is gynogamy? Transfer of pollen grains from anther of one flower to stigma of another flower but of same plant. That is known as zytogamy. Third one, xenogamy. What happens in case of xenogamy? Transfer of pollen grains from anther of one flower to stigma of another plant. From anther of one plant to stigma of another plant, that process is known as xenogamy. So, suppose this is a plant. Is this bisexual? It has bisexual flower, male part and female part. It is present on the same flower. It is monoecious. What is monoecious? When both male and female part they are present on the same plant. Monoecious, dioecious term is used for the plant. Unisexual, bisexual is used for flower. Monoecious, it means the plant has both male and female reproductive part. And if unisexual, bisexual is there, that means it is about the flower. Unisexual means it has only male or either male or female part is present. Bisexual means it has both the parts. Then, is it monoecious or dioecious? This part. It is monoecious. Why? Because although flower is unisexual, but both male and female they are present on the same plant. And when transfer of pollen grain from anther of one flower to stigma of same flower to stigma of different flower but of same plant, that is known as gynogamy. This gynogamy, can we say it as functionally xenogamy? Functionally it is cross pollination, but as it is the same plant, that's why genetically genetic makeup is same for both these flowers. That's why it is known genetically it is known as autogamy. Here, xenogamy. Are these two plants monoecious or dioecious? They are dioecious. Why? Because only one part is present on the plant, and if transfer of pollen grain from anther of one plant to stigma of different plant but of same species, that is known as xenogamy. Yes. Autogamy is self pollination or pollination within the same flower. If in case of dioecious plant only female part is present, that is known as pistillate. If only male part is present, that is known as staminate flower. If our flower is bisexual, that means flower is bisexual. It is also known as monoclinous. What is monoclinous? Both male and female parts are present on the same flower. Okay. In case of diclinous, what is diclinous? Either female flower is present or male flower is present. If female flower is present, that is known as pistillate. If male flower is present, that is known as staminate. Is autogamy possible in pistillate or staminate flowers? No. Why? Because autogamy is about the same flower only, and if only one part is present, then how it takes place? Then what about monoclinous ones? Monoclinous ones. In case of monoclinous, there are two chances. In bisexual flower, there are two chances that either these flower can be chesmogamous or clistogamous. Now, what is this chesmogamous and clistogamous? Chesmogamous, they are open flowers, whereas clistogamous ones, they are closed flowers. Closed flowers, and chesmogamous, they are open flower. In case of open flower, how autogamy can be possible? See, if both male and female parts, if they mature at the same time, that is homogamy. So homogamy should take place, then what happens? There are chances of autogamy in those plants. If both the male and female parts of the same flower, they mature at same time, then chances of autogamy are more. Second thing, there should be no obstacle between stamen and pistil. If there is obstacle, that condition is known as hercogamy. So hercogamy should not be there. Then what happens? Chances of autogamy can increase. Third thing is self incompatibility. What is self incompatibility? If same flower pollen grain reach to stigma of the same flower, then that grain does not allow to form pollen tube. That is self incompatibility. If this self incompatibility present, then autogamy doesn't occur, so it should be absent for autogamy to occur in chesmogamous or open flower. Now, clistogamous flowers. Clistogamous flowers, they are closed flowers, or we say these as invariably autogamous. Why? Because closed flowers, in them male and female parts, they are present within that flower, and they are, as they are closed, so maximum chances of autogamy are there. They provide assured seed set formation. Also, one more thing is there is no requirement of any pollinating agent. Why? Because these are closed flower. Male part and female part is present within that closed flower. So examples of some plants in which both chesmogamous as well as clistogamous flower is present. Viola oxalis commelina, they possess both chesmogamous as well as clistogamous flowers. These are clistogamous or closed flowers, which provides assured seed set formation. And these are chesmogamous, which are open flowers. Okay. Next, what are the advantages of clistogamy? Clistogamy has closed flowers, pure lines can be obtained, and homozygosity can be maintained. True breeding lines can be maintained in this case. As also one more thing, there is no dependency on the pollinating agent, and they provide assured seed set formation, guarantee of autogamy. Now, what are the disadvantages of these clostogamous flowers? Inbreeding depression can occur. What is inbreeding? Mating of closely related individuals is mating. Inbreeding. Then, due to that inbreeding, depression occurs. If autogamy continuously takes place, then what happens? Inbreeding depression occurs. As a result of this inbreeding depression, what happens? There are less chances of survival and variation. This is about chesmogamous, clistogamous flowers and advantages and disadvantages of clistogamous flowers. Now, autogamy, zygnogamy, xenogamy completes. Now we are going to start with agents of pollination. What are pollinating agents? Pollinating agents, they can be biotic and abiotic. In case of biotic, living things are used, living organism is used, whereas in case of abiotic, non-living one come into role. So in case of out of these biotic and abiotic, majority of plants use biotic agent for pollination. Abiotic agents, there are two, wind and water. Out of these wind and water, most common one is wind. If animal is used as biotic agent, then it is known as zoophily. If insect is used, it is known as entomophily. If bird is used, that is known as ornithophily. If bats are used for pollination, then that is known as chiropterophily. If ants are used, that condition is known as myrmecophily. If snail is used as biotic agent, then that condition is known as malacophily. If snake is used, then that condition is known as ophiophily. If wind is used as abiotic agent, and that is most common agent, then anemophily. If water is used as abiotic agent, then that condition is known as hydrophily. Majority of plants, they support biotic pollination agent, pollinating agent, but only small portion of plants use abiotic agent. Why? Because in case of abiotic pollinating agent, pollination is a chance factor. There can be pollination occur or no pollination. How that come to in order to compensate this, flower needs to produce large amount of pollen grain because pollen can be lost in this case. So in order to compensate that, flowers, they need to produce large amount of the pollen grains. In case of abiotic one, pollination is the chance factor. Now, what is anemophily? Anemophily is pollination by the wind, and wind is abiotic agent. What about the characteristics of pollen grain, stamen, stigma, ovule, flower in case of wind pollinating flowers? See, pollen grain, it should be heavy in weight or light in weight in order to easily disperse in wind current. They should be light in weight. Then are they sticky or non-sticky? They should be non-sticky so that they easily disperse in the wind current. Second part is stamens. Stamen, they are exposed so that they are easily dispersed. Pollen grains can be easily dispersed in wind currents. What about stigma of wind pollinated flower? Stigma should be feathery in order to trap these pollen grains. What about ovule? Single ovule must be present inside the ovary. What about flowers? If more than one flower or so many flowers are present, then they should be packed into an inflorescence. What is inflorescence? Arrangement of flowers on the floral axis. Arrangement of flowers on floral axis. If so many numerous flowers are present, then in that case they should be packed into an inflorescence so that they become visible. Next, ears. Example, one of the most common example of anemophily is corn cob. In case of corn cob, ears are there. What are these? They are female flower packed into an inflorescence. Stigma and style, they are wavy to trap the pollen grains. Example of Another example of anemophily is grasses. Here the picture shows compact inflorescence and well exposed stamen in case of wind pollinated flowers. Next is hydrophily. Anemophily complete, now hydrophily. What is hydrophily? Hydrophily is pollination through water. Examples of hydrophily. Water can be fresh water or marine water. Examples of the plants which pollinated with the help of fresh water, that is Vallisneria and Hydrilla. What about marine ones? Example is sea grasses or zostra. Now, there some another examples in case of those plants or flowers which are present inside the water but they doesn't take any help of water to get pollinated? Yes, aquatic plants which are not pollinated by water, not pollinated with help of water, these are known as water lily and water hyacinth. Then what are the pollinating agents among these water flowers? Water lily and water hyacinth, they get pollinated by insect or wind. Mark this is very important. Next, this hydrophily is rare in flowering plants and it is limited to 30 genera of monocotyledon. In most of water pollinated species, what happens? The pollen grain it protects from wet with the help of some covering. That covering is made up of mucilage. So in case of water pollinated species, pollen grains are protected from wetting by the mucilaginous covering. Hydrophily can be epihydrophily and hypohydrophily. What is epihydrophily? Epihydrophily is pollination takes place on the surface of water. That is epihydrophily. Example of that epihydrophily is freshwater plant, that is Vallisneria. In case of Vallisneria, what happens? Female flower reach to the surface of water. Female flower reach to surface of water with the help of a stalk. Then what about pollen grains? They are carried passively towards the surface. Example of epihydrophily is Vallisneria. Next, hypohydrophily. Hypohydrophily is pollination underwater. Example is seagrass or zostera. Here what happens? In as we see in case of epihydrophily, female flower reach to the surface of water with the help of stalk in case of Vallisneria. But here female flower it remains submerged inside the water. Pollen grains they are also released inside the water. In this case, pollen grain they can be ribbon like and they are carried passively inside the water. Now next, anemophily complete, hydrophily complete, that means abiotic agents done. Now biotic one, zoophily, pollination with the help of animals. And animals they are acts as pollinating agents, for example bees, butterflies, flies, ants, moths, birds, which bird, sunbird and hummingbird and bats, they are also used as pollination, we already discussed. Now, among animals, bees they are the insect in bees they are the dominant biotic pollinating agent. Mark this as very important. Next, larger animals like primates, arboreal, rodents, reptiles, and in case of reptiles, gecko lizard and garden lizard, they are act as pollinating agents. Next is entomophily. What is entomophily? When pollination is done with the help of insects, then that condition is known as entomophily. What are the specialty of those plants which are pollinated by insects? In these plants or flowers, they should be colorful. Why they should be colorful? In order to attract the pollinating agent or insect towards itself so that pollination can take place. Second thing, they should be rich in nectar. If flower is small, then what happens? So many flowers they can cluster into the inflorescence so that they becomes visible to that insect. Next, pollen grain. Pollen grain should be sticky so that they can stick to the body of the insect. When this insect come to another, goes to another flower for nectar, then what happens? Pollination takes place. Now, pollen nectar or robbers. They consume pollen or nectar without bringing pollination. They are pollen robbers and nectar. Pollen, yes, pollen robbers. Next, in order to sustain animal visit, flower need to provide some reward to that animal. What can be these rewards? First one is they can provide safe place for that biotic agent to lay eggs. One of the example is Amorphophallus, which is one of the tallest plant and it has 6 feet height. Then another relationship between yucca and moth. Yucca plant, scientific name of that yucca plant is Sonuba yuccacella. It only pollinated by its pollinating agent that is moth. They both shows mutualism together. Both gets benefited. They cannot complete their life cycle without each other. Both these species they get mutually benefited by this interaction. So moth it deposits egg in the ovarian locule of this yucca plant. As a result, this flower gets pollinated. Now, next topic is outbreeding devices. So before starting with outbreeding devices, let's solve some questions. Yes. Both wind and water pollinated flowers, they are not very colorful and do not produce nectar. What would be the reason for this? Because they do not require any such pollinating agents to attract. That's why they are not very colorful, no requirement of pollinating agent to attract. Clistogamous and chesmogamous flowers, they are produced by, example is Viola, Oxalis and Comelina. Majority of plants, they use which agent? Is it biotic or abiotic? Majority of plants, they use biotic agent for pollination. Zythnogamy. What is zythnogamy? Transfer of pollen grain from anther of one flower to another flower but of same plant. It is functionally xenogamy and genetically it is similar to autogamy because genetic makeup is of from same plant. Pollination that brings genetically different type of pollen grains to the stigma. Genetically different type of pollen grain can be bring with the help of xenogamy. Next, feathery stigma and well exposed stamens. This is a characteristic feature of which flowers which are pollinated with the help of wind, anemophily. Aquatic plants, they are not pollinated by water. What are those plants? Example, water hyacinth and water lily. Next, hypohydrophily is present in, what is hypohydrophily? Pollination inside the water. It is present in case of Zostera, seagrass. Next, pollen grains, they are non-motile. Is this statement true or false? True. Depending on the source of pollination, there are three types of pollination, that is autogamy, zythnogamy and xenogamy. In chesmogamous flowers, the anther and stigma, they lie close to each other. This is the statement true or false? This is about clistogamous flower, closed flower. Then dash flowers produce assured seed set even in the absence of pollinator. Those are clistogamous or closed flower. Next is zythnogamy. Is functionally it is known as xenogamy and genetically it is autogamy. Only type of pollination which brings genetically different type of pollen grain is xenogamy. Two abiotic and one biotic agent. Abiotic are wind, water. Biotic can be animals, and we already discussed about that. Majority of plants, they use which agent for pollination? Biotic. Pollination by dash is common amongst abiotic pollination. Pollination by wind or water? Wind. Next. Wind pollinators, they produce sticky pollen grain so that they stick to stigma. No, pollen grain should be non-sticky so that easily dispersed in wind current. Feathery stigma and well exposed stamen, they are feature of flowers which are pollinated with the help of wind. Wind pollinated flower, they have at least one number of ovule inside each ovary. Packed inflorescence is the feature of wind pollinator. Is this statement true or false? True. Example of wind pollination. Grasses, concave. Pollination by water is limited to how many genera of monocots or dicots? Monocots. 30 genera. Example of freshwater pollinated plant. Hydrilla. Example of marine water, zostera and sea grasses. Aquatic plants pollinated by water, they not pollinated by water are water lily and water hyacinth. Freshwater ones are Vallisneria, Hydrilla. In dash, epihydrophily is present. In case of Vallisneria, epihydrophily, that means above the surface of on the surface of water, pollination takes place. Next, next topic is outbreeding device. Yes. Next topic is outbreeding device. So as we see in case of flowering plants, majority of flowering plants, they are hermaphrodite flower. What is hermaphrodite flower? They are bisexual. Bisexual means both male and female present. Part present on the same flower. Then if both male and female parts they are present on the same flower, there can be chances, maximum chances of autogamy. But if continuously self-pollination or autogamy takes place, then what happen? It will result in inbreeding depression. Continuous autogamy, it results in inbreeding depression. What is inbreeding? Mating between the closely related individuals. If continuously autogamy takes place in these hermaphrodite or bisexual flowers, then what happens? Progeny it gets become weaker. There is can be no adaptation or no variations because genetic makeup is from the same plant only. Now, in order to avoid autogamy in case of these hermaphrodite flower, plant need to adapt something so that this autogamy can be discouraged and cross pollination or xenogamy can take place. So plant adapt some devices to promote xenogamy and to discourage autogamy. These devices are known as outbreeding devices. First one is dichogamy. What is dichogamy? If more both male and female parts they are present on the same flower, then how that autogamy can be prevented? See, if male part mature at some different time and female part mature at some different time, both male and female parts, if they mature at different time, then what happens? Suppose either pollen dehisces but stigma is not receptive, or stigma is become receptive but pollen grain not dehiscences. Then there are chances that autogamy cannot take place in this case. So there should be no synchronized or no coordinated manner work between pollen and pistil. Second, heterostyly. What is heterostyly? Suppose if filament and style they are having different length. If filament and style they are having different length, then how autogamy takes place in these? They are having different lengths, and there are no chances of less chances of autogamy. Although both male and female part they are present within the same flower. Third thing is, third outbreeding device is self incompatibility. What is self incompatibility? It is the genetic mechanism. If both male and female they are present on the same flower, suppose pollination takes place and pollen grain land on the stigma of that same flower, then what happens? That pollen grain does not able to fertilize the ovule. Pollen tube germination or pollen tube growth is inhibited. That condition is known as self incompatibility. Next, dicliny. What is dicliny? Production of the unisexual flower. If only one part is present, then there are also chances that no autogamy takes place. In case of sexuality of plant, plant can be monoecious, dioecious. What is monoecious? I am talking about plant. Then I use term monoecious and dioecious. Monoecious plant they are having both male and female part present within the same plant, not flower. Flower can also present but depends unisexual or bisexual. Okay, in case of dioecious, both the male and female parts they are present on the different plant. Example of monoecious one is, mark these as very important, castor and maize, both male and female parts they are present at the same plant. By seeing this diagram, can you please tell me in this case when flower is unisexual but plant is monoecious, out of autogamy, zythnogamy, xenogamy, which one is prevented? Autogamy is prevented. Why? Because only one part is present, one reproductive part is present in the one flower. But what about the zythnogamy? There are chances na that transfer of pollen grain from anther of one flower to stigma of the same another flower but of same plant. So here in this case monoecious, in case of castor and maize, autogamy is prevented, yes, but zythnogamy cannot be prevented. Next condition is dioecious. In dioecious, what are the example of dioecious? First, we need to remember papaya, date palm. Mark these as also important. Here in this case, is autogamy prevented? Yes, both male and female they are present on different plant now. What about zythnogamy? Zythnogamy is also prevented because both male and female they are present on the different plant. So dioecious prevent both autogamy as well as zythnogamy. This is about our topic outbreeding devices. So moving or switch to our next topic, that is pollen pistil interaction. Our next topic is pollen pistil interaction. Or this topic comes under post pollination development. We already discussed about pre fertilization event, pollination till pollination. Now post fertilization, post pollination, not fertilization, post pollination development. Pollen pistil interaction, it is the known as dynamic process. What is pollen pistil interaction? All the events which from landing of the pollen till its entry of pollen tube in the ovule. All the events which start from landing of the pollen to stigma till its entry inside the ovule. That is comes under pollen pistil interaction. So which part of the female recognize that pollen? Is it stigma, style, ovary or what? So pistil it recognize the pollen. Mark this is also very important. Pistil recognize the pollen. This pollen can be of right type or wrong type. We will see what is right type and wrong type. In case of wrong type of pollen, it either belongs to some other species or if it is from the same species but it is self incompatible, not able to germinate or form pollen tube, then also it is wrong type of pollen grain. In both these cases, what happens? That pollen grain will not germinate or its growth of pollen tube inside the female productive part is inhibited. What if right type of pollen grain lands on the stigma? That is also recognized by the pistil. If compatible pollen or right type of pollen lands on the stigma, it is from the same species, then what happens? There is some continuous chemical dialogue between pollen and pistil. With the help of this chemical dialogue between pollen and pistil able to recognize that either that pollen from the same species or from the different species or is it self incompatible. Then what happens if compatible pollen grain land on the stigma, then pollen germinates and pollen tube grows through the side. See, let's discuss about the flowchart of pollen pistil interaction. If compatible pollen lands on the stigma, then what happens? Pistil accept or reject? Pistil accept this pollen grain. There is continuous chemical dialogue between pollen and pistil. As a result of it, pollen accept this compatible pollen. Pistil accept this compatible pollen. Now, pollen starts to germinate and it will form pollen tube. From which part pollen tube is going to form? That germ pore where exine spore pollen is absent. What is the function of germ pore? Germ pore is the aperture where sporopollenin is not there. Aperture where sporopollenin absent is not there. This germ pore helps in the formation of pollen tube. Pollen tube, it grows through the stigma style and it will reach ultimately to the ovary. That pollen tube enters the ovule through which end micropyle and then enters which cell of that embryo sac, one of the synergid, and synergid has something filiform apparatus which guides the entry of this pollen tube. See, if compatible pollen lands on the stigma, then pistil accept it. Pollen starts to germinate. Pollen tube forms. Pollen tube grows through the style stigma and it will ultimately reaches to the ovary. Inside ovary, what is present? Ovule. Inside ovule, embryo sac is there. So this pollen tube enters into the embryo sac or ovule through which end micropyle, and out of these 7-celled 8 nucleus structure embryo sac, in which cell this pollen tube enters? That pollen tube enters in the one of the synergid cell, and synergid cell has filiform apparatus which guide the entry of this pollen tube. Now, what if question comes, in pollen tube is vegetative cell nucleus comes first or male gamete? So by seeing this diagram, we can say that male gametes are entering first and then vegetative nucleus is there. Remember these labelings also. Now, how many male gametes are there? Two male gametes. In case of 60% angiosperm, we already discussed that pollen shed at how many cell stage? Two cell stage. What are these two cells? Vegetative cell and generative cell. Then what about in this case, how two gametes are formed? Generative cell, it will undergoes mitosis during the pollen tube growth in the stigma. During this pollen tube growth in the stigma, what happens? This generative cell undergoes one mitosis division to form two male gametes. This is about pollen pistil interaction. Next topic is artificial hybridization. Artificially hybrids are formed. We'll see how. So some plant breeder, they are interested in crossing some different species to combine their desirable traits or characters so that they will get some superior varieties. How this can be achieved then? See, this can be achieved with the help of technique named artificial hybridization. Artificial hybridization, it is the one of the major approach for these crop improvement program. One thing need to be remembered that only desired pollen grain are used for pollination And stigma, it is also protected from the contamination from unwanted pollen grain. So what is artificial hybridization? What are the steps? We'll see. Suppose if female, which if I choose some female parent plant, then female parent plant, there are chances that that plant which whose flower I choose as female parent, then whose flower I choose as female parent. So there are both male and female parts are present in this flower. How I prevent autogamy here? How I prevent pollination of the same transfer of the same pollen grain from anther to stigma of the same flower? These pollen grains needs to be removed. These pollen grains needs to be removed or cut. So first step here is, if my female parent plant is bisexual flower, has bisexual flower, then first step is emasculation. What is that emasculation? Removal of these male reproductive parts with the help of forceps. That female, that male part stamen is removed with the help of forceps. That process is known as emasculation. Okay, autogamy is prevented by the emasculation. What about xenogamy? Or what about xenogamy? Cross pollination. There are chances that this some another pollen grain can also land on the stigma. How that can be prevented? See, second step is bagging. That emasculated flower from where anthers are already removed. That emasculated flower then cover with the bag. Emasculated flower covered with the bag, and that bag is made up of butter paper. If I cover that female flower now with the help of bag, then cross pollination also prevented. In order to avoid contamination from unwanted pollen grain, I only want which the pollen grain which I selected to pollinate here. Then what happens? In order to avoid that contamination, that emasculated flower is covered with the bag. Third step, if stigma of that flower retains attains receptivity, then transfer of the desired pollen grain over here. And fourth step is rebagging. Pollinated flower is rebagged so that post fertilization or post pollination event can occur. Okay, this condition is about my female parent plant, in that flower is bisexual. What if female parent has unisexual flower? Only one, only female reproductive part is there. If only female reproductive part is there, then is emasculation required here? There is no requirement of emasculation. No requirement of emasculation, because only female reproductive part is present, anther is not present, semen is not present. Second thing, second step is bagging. No emasculation, only bagging is required in order to avoid contamination from unwanted pollen. Only desired pollen can transfer if stigma retains receptivity. If stigma attains receptivity, then desired pollen transferred and flowers they are again rebagged. This is about artificial hybridization technique. Now our next topic is double fertilization. Before starting with that, let's solve some questions. Yes, let's recall our topic which we already discussed now. Dichogamy and heterosily prevents what? They prevent inbreeding. These are outbreeding devices. Self-incompatibility, it is a genetic mechanism that prevents self-pollen from fertilizing the ovule by inhibiting pollen tube formation or pollen tube growth inside the pistil. One of the tallest flower, it is about how many feet? 6 feet, and its name is Amorphophallus. Continued self-pollination results in inbreeding depression. Papaya is monoecious or dioecious? It is dioecious. Both male and female, they are present on the different plants. Yucca and dash complete their life cycle without each other. Yucca and moth. Pollen grains, they are long and ribbon-like. Is it case of epihydrophily or hypohydrophily? This is about hypohydrophily. Next. Which of the following plants they produce two types of flowers, desmogamous as well as clistogamous? Three are there, Viola, Oxalis and Commelina. This. What is the term used to describe the type of flower that do not open at all? These are clistogamous flowers. Clistogamous. Next. Why is there no chance of cross pollen landing on stigma in clistogamous flower? Because we know the flower is closed in case of clistogamous flower. Next question. The largest cell of mature embryo sac is? It is central cell which has two polar nuclei. Next question. Which of the following plant provides safe place to lay egg? One of the large tallest flower, that is Amorphophallus, and its height is about 6 feet. Next, which of the following is not a characteristic feature of insect pollinated flower? Fragrance, fall odor, nectaries, mucilaginous covering or pollen grain. Mucilaginous covering is in case of hydrophily. Pollen grain is protected from wetting with the help of mucilaginous covering. Next, dioecious condition prevent. Dioecious means both male and female, they are present on the different plants. It prevents autogamy as well as zythnogamy. Autogamy and zythnogamy. We already discussed. Zythnogamy is functionally autogamous, genetically allogamous, ecologically autogamous or genetically autogamous. Functionally it is xenogamy. Genetically it is autogamy because transfer of pollen from anther of one flower to stigma of another flower, but that flower belongs to the same plant. Next question. For outbreeding devices we discussed self-incompatibility, heterostyle, dichogamy. Next, monoecious examples. Example of monoecious is castor, maize. In two-cell pollen, the generative cell divides during growth of the pollen tube in stigma. Dash guide the entry of pollen tube, filiform apparatus. They guide the entry of pollen tube. What is emasculation? In case of female parent plant, if the flower is bisexual, the stamen is cut with the help of forceps in order to avoid self-pollination. Emasculation flowers, they are covered with the help of bag made up of, that bag is made up of butter paper. This process is known as bagging. There is no need for emasculation in which flower if female parent flower is unisexual. In some species, pollen release and stigma receptivity is not synchronized. It is example of outbreeding device. Pollen robbers or nectar robbers. They consume pollen or nectar. The number of mitotic generations required to form a mature embryo sac in flowering plant. Three mitotic divisions occur in functional megaspore. Three. Next, the devices to discourage self-pollination. The devices to discourage self-pollination are pollen release and stigma recept is not synchronized. Anther and stigma they place at different position. Rejection of pollen by stigma of cell flower, that is self-incompatibility. All of these are example of outbreeding devices. In monoecious plants like castor and maize, what happens? Autogamy is prevented, but zypnogamy is not prevented because same plant has both male and female reproductive part. Next, pollination in water hyacinth and water lily is brought about by the agency of, is it water? No, insect or wind. Next, attractants and rewards are required in which case? Anemophily? No. Entomophily? Yes. Pollination through insect. Hydrophily? No. Cleistogamy are closed flowers. Flowers which have single ovule in the ovary and they are packed into inflorescence, they are usually pollinated by wind poll. These are the characteristics of wind pollinated flowers. Next question. Emasculation. It prevents self-pollination in female parent. It prevents cross-pollination in female parent. It prevents self-pollination in male parent. It prevents cross-pollination. Which emasculation prevents self-pollination in the female parent. Okay. So moving towards our next topic, that is double fertilization. So we'll start with the topic after a short break of 2 to 3 minutes. Okay. So our next topic is double fertilization. This is the structure of embryo sac. We already discussed about this. Pollen tube. It has vegetative nucleus and two male gametes. This is micropyle and thalassal end. Towards thalassal end, there are three antipodal cells. Large central cell, then pollen nuclei. They are present in the central cell. Egg apparatus. It includes synergids plus egg cell. And synergids have filiform apparatus which guides the entry of this pollen tube. Now, next process is double fertilization. What happens? How many male gametes are there? Two male gametes. They are formed by division, mitotic division of generative cell. Two male gametes are there. Out of these two male gametes, one pollen tube enters in the one of synergid cell. Then what happens? Synergid degenerates. So one male gamete fuse with the egg cell. One male gamete fuses with the egg cell, and after fusion, it forms zygote. That process, when one male gamete fuses with egg cell, that is known as syngamy. Next, another male gamete fuses with the two polar nuclei which are present in the central cell. Another male gamete fuses with these two polar nuclei. They are N, and another male gamete is also N. So what is going to form? Three nucleus are fused. That's why this fusion is known as triple fusion, and it will form primary endosperm nucleus. As a result of syngamy, zygote is formed, and as a result of triple fusion, primary endosperm nucleus is formed. This syngamy and triple fusion, syngamy plus triple fusion, both will together make double fertilization, and this double fertilization is one of the unique event in case of angiosperm plant. This is about double fertilization. Now we'll see about fertilized embryo sac. This end is thalassal end, that end is micropyle end. Towards thalassal end, initially antipodal cells are present, but after fertilization, what happens to these antipodal cells? These cells will get degenerate. Then, what about micropyle end? Towards micropyle end, egg apparatus, synergid plus egg cell is present. After syngamy with one egg cell and male gamete, diploid zygote is formed. That zygote going to further develops into embryo. What happened to synergids? Synergids, they also get degenerate. What about here? Initially, polar nuclei are present. When these polar nuclei, they will get fused with the male gamete. Triple fusion takes place, and as a result of it, primary endosperm nucleus is formed, and that primary endosperm nucleus PEN is triploid 3N. Then what about the central cell where these primary endosperm nucleus is formed? Initially, this is central cell, but after triple fusion, this central cell changes to primary endosperm cell. This is about fertilized embryo sac. Now, post-fertilization changes. What happens to ovary? Ovary after double fertilization, it will become fruit. What about ovary wall? Ovary wall, now it becomes wall of the fruit wall. and fruit wall is pericarp. Next, what about ovule? Ovule form seed, funicle. Initially it is the stalk of ovule. Now it will become stalk of seed. What happens to nucellus if nucellus persistent after double fertilization? That nucellus become perispum. And this condition is seen in case of black pepper and beet. What about outer integument? Initially, this outer integument is the outer covering of ovule. Now it will become outer covering of seed. Seed coat, outer seed coat. And outer seed coat is known as testa. What about inner integument? Initially, this inner integument is the protective covering of ovule. Now it will become protective covering of seed and it is inner seed covering that is known as tegmen. Next, micropyle. Initially, micropyle is that region where these integument layers are absent. Now, after double fertilization, this micropyle it appear as a pore which facilitate the transport of gases during the germination of seed. Fertilized egg, zygote. Zygote going to form embryo. Then synergids. What happened to synergids? Synergids become degenerate. Next, primary endosperm cell. Primary endosperm cell will going to give rise to endosperm which is nutritive tissue and which will going to provide nutrition to the developing embryo. What happens to antipodal cells? Antipodal cells they also become degenerate. Next, endosperm. So basically what happens? Endosperm it develops from the primary endosperm cell. Can you please tell me why the endosperm development precedes embryo development? Because this endosperm is nutritive tissue. This endosperm is nutritive tissue. That means which is going to provide nutrition to this developing embryo. That's why development of this endosperm precedes the development of embryo. So this endosperm development it can be free nuclear or cellular. That primary endosperm cell mitotically divides and give rise to endosperm that is nutritive tissue which going to provide nutrition to the developing embryos. So endosperm development, it can be free nuclear. That means cytokinesis immediately does not follow karyokinesis. Karyokinesis is not followed by cytokinesis. Only karyokinesis takes place, and it can be cellular when cell wall laid down. So we need to learn the examples here. In case of free nuclear, example is coconut water. The coconut water which we drink, that is the example of free nuclear endosperm. This is free nuclear endosperm. And what about cellular one? When cell wall laid down, then that becomes white kernel part of the coconut. So white kernel part of the coconut is the example of cellular endosperm. Next, this endosperm is utilized by the developing embryo as a nutrition. So this endosperm, there are chances that this endosperm can be completely consumed by the developing embryo, or chances are also that this endosperm not completely consumed by the developing embryo. So if if that endosperm is completely consumed by the developing embryo, then that condition is known as non-endospermic. It is also known as non-albuminous, or it is also known as X-albuminous. When that endosperm is completely utilized by the developing embryo, then that condition is known as non-endospermic, non-albuminous, X-albuminous. So example of the plant where this non-albuminous condition is found is dicot. An example is pea, bean, groundnut, sunflower, where this endosperm is completely utilized by the developing embryo. But exceptions are there, and these exceptions are given in Morphology of Flowering Plants chapter. Can you please tell me what are these exceptions? In which dicot that endosperm after still persists by the, or it is not consumed by the developing embryo? So example is castor. Castor is one of the dicot in which endosperm still persists. Next case is when endosperm is not completely utilized by the developing embryo. That condition is known as endospermic. That means endosperm is there. Example is monocot and coconut, wheat, maize. But here also exception is there. Monocot, where that endosperm is completely utilized. Endosperm is completely utilized. That example is orchid. Here castor is dicot where endosperm still present. Endosperm is there. Next, dicot embryo. So dicote embryo, it develops from zygote. It will give rise to embryo. Zygote is 2n. Next, most zygote it divides only after certain amount of endosperm is formed. Why is it so? Because endosperm is nutritive tissue which is going to provide nutrition to developing embryo. That's why zygote divide only after certain amount of endosperm is formed in order to get the nutrition from the endosperm. Stages of embryogeny in dicots is first pro-embryo is there, then globular embryo, then heart-shaped embryo is formed, and last one is mature embryo. We'll discuss about these in upcoming slides. So embryo it consists of, it has embryonal axis. We'll see what is embryonal axis, and it has cotyledon. This is about dicot embryo. Globular embryo, then heart-shaped embryo, then mature embryo. We'll see what how these are formed. Stages in embryo development in case of dicot. We'll see in the next slide. Stages of embryogeny in dicot. Initially, what is present? Zygote. Zygote is diploid. This end is telasal end, that end is micropyle end. What happens? Unequal division or transverse division occurs in that zygote. Mitotic division occurs. Because of that, what happens? Two cells are formed, unequal cells. The cell which is going to form toward micropyle and that cell is known as basal cell, and the cell which is going to form towards telasal and that cell is known as terminal cell. Then what happens? The basal cell which is present towards the micropyle, that basal cell undergoes transverse division. Transverse division and it will form suspensor. This is terminal cell, and the basal cell when it undergoes transverse division then it will form suspensor. What is the role of this suspensor? This suspensor help in pushing that embryo in the endosperm. So topmost cell of this suspensor it becomes swollen and that cell is known as haustoria. The lowermost cell of this suspensor is known as hypophysis. Then what happens? Initially basal cell which is present towards micropyle and that divides. Now terminal cell divides. Terminal cell divides to form proembryonal octet. This terminal cell now divide to form proembryonal octet. Proembryonal octet is formed towards the telasal end. This is about the proembryo. This Hypophysis of suspensor, it helps in formation of root cap and root cortex. Uppermost cell of this proembryo, they are going to form hypocotyle, and lower four cells are help in formation of plumule and cotyledons. Then what happens? That proembryo going to form heart-shaped embryo. That proembryo is going to form heart-shaped embryo. Same suspensor, topmost cell is Hostoria. This is heart-shaped embryo, and heart-shaped embryo has cotyledons and plumule. Then this heart-shaped embryo going to form mature embryo. Mature embryo also has cotyledons, plumule, embryonal axis. We'll see in next slide. Yes. This is about mature embryo. Mature embryo has suspensor. Here it has radical, plumule and two cotyledons. This is about mature embryo in case of dicot. Yes. Now dicot embryo. It has plumule. What is the role of plumule? It helps in shoot formation. Then these are two cotyledons in case of dicot. Cotyledons, they contain reserve food material. There are two cotyledons in case of dicot. Next, hypocotyle. Hypocotyle is the portion of embryonal axis which is present below the level of cotyledon. Portion of embryonal axis which is present below the level of cotyledon. Then what is epicotyle? This is the level of cotyledon, and epicotyle is the that region which helps in formation of plumule. Epicotyle, it is the portion of embryonal axis which is present above the level of cotyledon. So it is present above the level of cotyledon. Epicotyle above the level, hypocotyle below the level. How many cotyledons? Two cotyledons. Plumule help in shoot formation. Radical. Radical helps in root formation. And that radical is covered by the root cap. This is about dicot embryo. How that dicot mature embryo form will already discuss. Now, monocot embryo. In case of monocots, they are having only one cotyledon that is known as scutellum. Scutellum is present towards the lateral side of embryonal axis. Towards lateral side of embryonal axis. What about the shape of scutellum? It has shield shape. Mark this as very important. It is given in morphology of flowering plants. It has shield shape. And that one cotyledon is known as scutellum. Now embryonal axis. Embryonal axis has plumule. Plumule covering of plumule is coleoptile. Coleoptile is undifferentiated sheath around the plumule. Then epiblast. Epiblast is here in case of monocot, there is only one cotyledon, but this epiblast represents the remnant of second cotyledon, or it is rudimentary cotyledon. Then radical. Radical helps in root formation. Coleorhiza is the undifferentiated sheath around. Radical. R for radical, R for rhiza, coleorhiza. Coleoptile is foliar structure, leaves like undifferentiated sheath is coleorhiza. Foliar structure around the plumule. It is plumule covering. This is about monocot embryo. Now seed. Final product of sexual reproduction is seed. Ovule after fertilization it develops into seed. Seed it consists of seed coats, cotyledon, and embryonal axis. Seed coats, there are two types, two seed coats. Outer seed coat is known as testa and inner one is known as tegument. Integuments, outer integument, inner integument, they will become seed coat. Now cotyledon, it contains reserve food material which provide nutrition to developing seed. Reserve food material. Perisperm. What is perisperm? If nucellus persists, then that is known as perisperm. Persistent nucellus. Example of perisperm is black pepper and beet. In these cases, nucellus still persists. Micropyle after fertilization. What happens to micropyle after fertilization? It appears as a pore which facilitates entry of or exchange of gases and it also allows entry of water. Seed maturation. When seed matures, then what happens? It's 10 to 15% water content gets reduced. Then seed becomes non-functional or it will undergoes into dormant stage. Whenever favorable condition will be there, then this seed starts to germinate. 10 to 15%. Here, by mass. It will lose 10 to 15% moisture by mass. That seed enters in inactive stage, that is dormant. Whenever favorable condition will be there, that seed starts to germinate. Now, here is the picture represent seed of some plant. Yes. This is about onion. You can see endosperm is there, seed coat, cotyledon, shoot apical meristem, root tip, hypocotyle root axis. Then what about this diagram? It shows castor seed. Castor is one of the dicot which is having endosperm. That endosperm is not completely utilized by the developing embryo. So endosperm is there, cotyledon, seed coat. You can see the labelings. This is about maize. In case of monocots, what happened? Endosperm is the endosperm is there. That endosperm differentiate from the embryonal part with the help of or this endosperm separate from this embryonal axis part with the help of this layer. That layer is made up of protein and this layer is known as alluron layer. This is alluron layer. In case of monocots, they are having scutellum which is present on the lateral side of the embryonal axis. This scutellum has shield shape and embryonal axis has plumule radical. Coleoptile is the foliar-like structure around the plumule. It has leaf primordias. Radical, it helps in shoot formation and undifferentiated sheath around radical is coleoptriza. This is about the seeds of some plant. Now, advantages of seed to angiosperms. Seed, as they are the result of sexual reproduction. These are the basis of agriculture. Being product of sexual reproduction, they are going to provide new genetic combinations, new variations. The seed coat, hard seed coats, outer is testa, inner is segment, they protect the young embryo. Seed, they are having better strategies to for dispersal to new habitats and reproductive processes like pollination and fertilization. They are independent of water, but seed formation is more dependable as they are having sufficient food reserve. So young seedling they are nourished until they are capable of photosynthesis on their own with the help of cotyledons. Now seed viability. Seed viability, it is the time period in which seed retains its ability to germinate. Several records of old but viable seeds are there. The one of the oldest example, one of the oldest is of lupine. Scientific name of that lupine is Lupinus arcticus that is excavated from the Arctic tundra. This seed germinate and flowered after an estimated record of 10,000 years of their dormancy. And a recent record of 2000-year-old viable seed is of the date palm and scientific name of date palm is Phoenix dactylifera that is discovered during excavation at King's Herod Place near the Dead Sea. Now next topic is fruit. So ovary after fertilization, ovary develops into fruit. Wall of ovary develops into the fruit wall and fruit wall is known as pericarp. If that pericarp is thick and fleshy, if pericarp is thick and fleshy, then it will further divided into three parts: epicarp, endocarp, and mesocarp. Epicarp, endocarp, and mesocarp. Fleshy fruits. Examples of fleshy fruits, they are orange, what about dry fruits? Example is groundnut. Types of fruit. Fruit can be divided into false fruit and true fruit. What are true fruits? When fruit develops from the ovary, then they are known as true fruit. What about false fruit? When fruit develops from part other than ovary, then it is known as false fruit. An example of false fruit is apple, strawberry, and cashew. This is about false fruit and true fruits. When fruit develop from the ovary, they are true fruits. When they are developed from the parts other than ovary, then they are known as false fruit. Now, parthenocarpic fruit. Parthenocarpic fruit. When fruit forms, formation of fruit without fertilization is known as parthenocarpic fruit. Without fertilization, they are seedless fruits. And formation of fruit can be by inducing hormones, induced with the help of hormones. Example of parthenocarpic fruit is banana. So this is about fruit. Next and last topic of this chapter is apomixis. What is apomixis? When formation of seed without fertilization. Seed forms without fertilization is apomixis. When fruit form without fertilization, that condition was parthenocarpy. Then what about formation of organism without fertilization? That condition is known as parthenogenesis. An example of parthenogenesis, we already discussed that about in sex determination topic. Drone. They are formed with the help of parthenogenesis. Here, formation of seed without fertilization. Example where apomixis is seen is Asteraceae family, sunflower family, and grasses. Then this apomixis is the form of asexual reproduction, but it mimics sexual reproduction. How it mimics sexual reproduction? Because seed is formed and seed is the result of sexual reproduction. Here there is no meiosis division. That's why we are saying it is a form of asexual reproduction which mimics or copy sexual reproduction. One of the most important question, that is how these apomictic seeds are formed. How seeds are formed without fertilization. In our syllabus there are two steps or two parts. We'll see apomixis. It can be non-recurrent, recurrent apomixis, and third one is adventive embryogeny. What happen in case of recurrent apomixis? There is no meiosis division and embryo sac is formed without this meiosis division. Ovule has three parts: integument, nucellus, megaspore mother cell. Inside it, these three parts are present. Integument is diploid, nucellus it is also diploid, megaspore mother cell is also diploid. When these two parts, integument and nucellus, they are going to form embryo sac, then that condition is known as apospory. But what if this megaspore mother cell, if megaspore mother cell going to form the embryo sac without meiosis division, that condition is known as diplospory. We need to remember this, diplospory. What happens? Ovule, inside ovule megaspore mother cell is there. That megaspore mother cell undergoes mitosis division and it will form embryo sac without meiosis division. Then that embryo sac has egg which divides to give rise to embryo. So this is about recurrent apomixis. Next we need to learn is adventive embryogeny. What happens in this case? Ovule has nucellus, integument, they encircle inside themselves nucellus. Nucellus is diploid, that nucellus divides and it protrudes inside that embryo sac that develops into embryo, and this condition is known as adventive embryogeny. When nucellus without any meiosis division, it protrudes into the embryo sac by mitosis division. Example of this is mango and citrus. Here in this case, more than one ovule is or more than one embryo is found. That condition is known as polyembryony. So after apomixis, seed is formed. Okay. Are these clones? Are they genetically similar to plant? Yes, they are genetically similar to plant because no meiosis division occurs. Meiosis leads to segregation of character. That's why we are saying they are the plants which formed after apomixis. They are clones, genetically similar to their parent plant. These are two methods how apomixis or how apomictic seed can formed. Remember this thing that nucellus without meiosis division, it will protrude into embryo sac. It divide mitotically to give rise to embryo. So many embryos are present inside that One ovule, so that condition is known as polyembryony. Many ovules, many embryos inside single ovule. That condition is known as polyembryony, and polyembryony is seen in case of citrus and mango. So let's start to solve some questions. Chapter finish. Yes. So let's recall. Dash is an event unique to angiosperm, that is double fertilization. Suspensor, it grows to push embryo away from which end? Away from the micropyle end. It helps in pushing the embryo towards the endosperm. Embryo development precedes endosperm development. Is this statement true? No. Endosperm develop first because to assure the nutrient supply to this endosperm. This statement is incorrect. Pre-nuclear endosperm example. The liquid part of coconut. Endosperm is completely consumed in case of some dicots, but exceptions are there. We already discussed. Endosperm, it persists in mature seeds in monocots, but some exceptions we already discussed. The zygote give rise to embryo and subsequently proembryo is formed, then heart-shaped embryo, then mature globular heart-shape and mature. Embryo, it consists of embryonal axis and cotyledons. Embryonal axis, above the level of attachment of cotyledon, above the level is epicotyle, below the level is hypocotyle. Non-albuminous seed does not have endosperm. Residual persistent nucleus, it is called as perisperm. It is found in case of black pepper and beet. Albuminous seed examples we already discussed. Integument develops into seed coat. Testa tegmen. This facilitate the entry of water and oxygen for germination. Micropyle, which appears as a pore. Parthenocarpic fruit, formation of fruit without fertilization. These are seedless fruit, and example is banana. They are induced by giving hormones. Example of false fruit. False fruits are those fruit which develop from the parts other than ovary. And example of false fruits we already discussed: strawberry, apple, and cashew. Apomixis, it is present in Asteraceae family and grasses. Apomixis, it is a form of sexual reproduction which mimics asexual reproduction. Is it true or false? It is a asexual reproduction form, but it mimics sexual. Polyembryony examples are mango. This is about sexual reproduction in flowering plants chapter. That's all for today's class. Thank you so much.